our new equipment that we're testing out, but so far so good. Everything looks like it's working. So welcome to our speaker series, our quarterly speaker series that we do at Save the Great South Bay. My name is Todd Shaw. I'm president of the organization, we just love this format of being able to have people chat with each other. Maybe you haven't seen each other before, or you're just reconnecting, or you're making new friends. It's just a great way to start the day. We have an amazing program today. It is our 5th annual State of the Bay with Dr. Christopher Gobler, and it's always very, very informative, and it's really kind of an annual event that we really look forward to every single year. So with that, I'd like to introduce Robin Silvestri, our Executive Director, to kick off the session.
Thank you. Thank you, good morning, everybody. Can you hear me okay? Because sometimes we have a difficulty. Can you hear me in the back of the room okay? Okay, great. Well, thank you so much for joining us. As Todd said, this is our 5th annual State of the Bay speaker series with Dr. Gobler. We're always very honored to have him here to explain the science of the water quality data to us and the current state of the environmental events happening out on the Bay. Before I get started, I just want to acknowledge a few people who are in the room with us, including our board members, Rain Horsley, Todd Shaw, Brianna Quinradi, and Andy Rochelle. They are dedicated volunteers, so let's give them a little round of applause. I'd also like to recognize our creek defenders who are out in the audience. These guys come out, and ladies, come out and defend the 50 creeks that lead into the Great South Bay between Massapequa and Mastic Beach. So today in the audience we have Walt Meschenberg for Mastic, Ed Reagan, who runs our Day in the Life program, Todd, who runs Babylon Carl's River, and Andy Michelle, who does Sumpalms Creek over in West Islip, and of course Mayor Siri, who takes good care of the creeks over in Amityville. Nothing happens here or anything that we do without our volunteers, so thank you to Amy, Kathy, and Kelly for coming out this morning to help us set up. Ed was a big help in setting up our new PA system. I'd also like to recognize, thank you, Renata. I also want to recognize that everything that we do is a group effort. Nothing happens in a silo if we want to move the needle on improving water quality in the Great South Bay. And that includes working together with many of our local public officials to secure permitting and funding to help us complete our project. So I'd like to recognize Legislator Mazzarella, who recently helped us secure an $80,000 grant to do an invasive removal out in Mastic Beach. Congressman Garbarino is represented today. Senator Wake, we have Bill Doyle joining us today, and we have Mayor Siri from the village of Amityville. We work with all levels of government, from the governor's office down to our local village mayors. This is a really important part of our structure. In addition to that, we have a lot of our partners that we work together with on different projects with us today, including the Nature Conservancy, Seatuck Environmental, Cornell Cooperative, the South Shore Estuary Reserve is represented today. DEC, our favorite permitting guys, are in the house. Coastal Research and Education Society of Long Island, Suffolk County Department of Health, who's been a key partner us in water quality monitoring this year. Keep Islip Clean, Save Environmental, which is very active in eelgrass restoration, and we always want to recognize our oyster farmers who are putting millions of oysters back in the bay for commercial purposes. But those oysters, while they're in there, provide a really great environmental function of filtration. So thank you to Sixto Portilla, who brings his kids out. Sixto, if you can make your family stand up, and I just want to recognize that you bring your kids to these events. Sixto runs Mars Bella Oyster Farm, and he's a great partner for us. So with that, I would like to introduce Dr. Gobler and give you a little background on him. Dr. Gobler is, of course, a professor at Stony Brook School of Marine and Atmospheric Sciences. He has received his MS and his Ph.D. from Stony Brook, and he began his academic career at Long Island University in 1999. In 2005, he joined Stony Brook University as the Director of Academic Programs for the SoMAS in the Southampton campus. He was born and raised here on the island. He grew up enjoying swimming and fishing and sailing, and his pursuit in his education was motivated by what we all know, the progressive declines in Long Island shell fisheries, the decimation of the clam populations right here, which really changed the landscape of the Great South Bay. During the past 20 years, his research has identified the key role of excessive nitrogen. If you've been to any of our speaker series in the last 5 years, you know we're always talking about the nitrogen coming from the cesspools off of Long Island. With the Clean Water Center for Clean Water Technology, Dr. Gobler is developing alternatives to cesspools that are environmentally friendly. We're really excited to see how that progresses. I believe you're in implementation stages with those at this point. So Dr. Gobler sees the promise of discovering the solutions to Long Island's nitrogen problems, as well as the creation of an industry that can create jobs for Long Island. And with that, I'd like to hand the mic over to Dr. Gobler.
Okay, well, thank you so much, Robin. Thank you to Save the Great South Bay for the invitation. And thank you for everyone for coming out. It's good to see so many friendly faces, and it's great to share the information. Okay, I'll start just by acknowledging the fine young men and women who work in my lab and really are responsible for all the data you're going to see today. They're the boots on the ground and fins in the water making this happen. University students and technicians and postdocs and such. Okay, and the first concept we'll cover is one we're all familiar with, and the idea that Long Island is a watershed, and you can't escape it. No matter where you are, the water that's falling on land is going to eventually have two very important impacts on actually all of our lives. That water, one, is our source of drinking water, and so anything that's happening on land will affect that water, and that's eventually what comes through our taps. And more, if you're following what's happening with regards to bottled water and microplastics, you should rely on that tap water as your drinking water source. But beyond that, if we're not extracting it to drink, it's discharging into our surface waters, and it's very clear in that regard it can have a very strong effect on surface waters and coastal ecosystems. So the trends with regards to the population of Suffolk County and the amount of nitrogen in the groundwater below Suffolk County are pretty clear. Suffolk County is now the third largest county in all of New York State. I'll get back to that momentarily, just behind Brooklyn and Queens, not just behind Brooklyn and Queens, well behind. But as that population grew, so did the levels of nitrogen in our groundwater, and to the point where we've got an average of about 3.8 milligrams per liter. And in a national perspective, if you look at this graph here, this is, if you look at the y-axis, it's in hundreds of millions of people. 200 million is that first bar. So this is the water supply for Americans. We have a population of about 330, 340, 350 million people in the US. So 2/3 of the country have drinking water with less than 1 milligram per liter. So we're kind of at the tail end with higher levels. In some areas, if you look at this heat map of where the levels of nitrate are high and low, are even higher than that. And I won't spend a lot of time on this, but I just will point out that nitrate in drinking water is not something that's beneficial. There's a standard by the EPA of 10 milligrams per liter, and we don't exceed that. But there is increasing epidemiological research in the last decade that has shown that levels below that have been linked to cancers and adverse birth outcomes. And so we want those nitrate levels to be as low as possible. And it's maybe not a coincidence that very recently, I think it was just not even a year ago, the EPA is now reinvestigating what the standards should be for nitrate in drinking water because of this steady drumbeat of all these findings of linkages between nitrate and adverse health outcomes. But I'm a marine scientist doctor, not a medical doctor, so let's get into the bays here. You're the first public audience to see this map. Or there's maybe, I think, only one other human on the planet who's seen it up until now. So this is the water quality map that we've generated for the summer of 2024. And if you've seen my prior maps, it's actually not all that different, but essentially it's indicating the areas of low oxygen and harmful algal blooms for all of Long Island, as you can see, all the way from the city all the way out to Montauk. And so what it shows is actually a record number of dead zones that we detected in this past summer. I'll get more back to that later, but those are areas of low oxygen. We've never seen this many, this number, more than 36 different individual locations with low oxygen. With regards to freshwater harmful algal blooms, also a near record number of more than 24 of these. And it also depicts more than 24 different harmful algal blooms in marine ecosystems, as well as fish kills in the South Shore, all of which were coincident with low oxygen zones. So to talk about some of these harmful algal blooms, there's two general classifications, those that are toxic to humans and animals, and those that we call ecosystem disruptive. Today I'm really just going to focus on the ones that make biotoxins and therefore are a threat to human and animal health. In either front, we've done a lot of research over the last decade, 2 decades, making strong links between higher levels of nitrogen and these blooms getting more intense, but also them becoming more toxic. More nitrogen can lead to more toxicity. I'll probably emphasize that later. I'll start with the inland blooms. These are sometimes known as cyanobacteria or blue-green algae blooms. And if you're not familiar with the site, that we're looking at there is not the green of a golf course, but that's a lake. That's Lake Agawam in Southampton. And we worry about these blue-green algal blooms because they do make toxins. For example, the most common toxin associated with these blue-green algae blooms in New York on Long Island is called microcystin. When it was first identified, that toxin was called fast death factor until they came up with the actual name. It's a danger to human health, has been associated with cancers. Strong enough to kill elephants. This was published just a few years ago in Africa, and has been associated with hundreds of dog poisonings across the U.S. There's a paper by the CDC came out not long ago. And DEC has been monitoring these and looking very carefully at these. Now, I should say this map here is total number of water bodies. So this isn't representing that they're getting worse, frankly. This in particular is just what I would call a great awakening, and that the state recognizing that this is a widespread problem. But what's interesting is if you look at this map, and I know the screen, particularly with people in the back, is small, so I'm going to try to give a narrative, so you don't need to see the details. But this is a graph essentially showing 48 different counties in New York. If you don't know, there's 62 counties total. So in 2023, 48 of those counties had blue-green algae blooms, but no county had more than Suffolk County. And so the number in 2023 was at 23 sites. We had 24 this summer. It always comes in around there, in fact, but more than double the nearest county. And in 2024, I can't do the full state listing yet because I don't have all the data from the state. But what I can say is on the South Fork of Long Island in particular, there were 17 of these events alone. So it's really a South Fork problem associated, I shouldn't say it's not exclusively a South Fork problem. This is just a 2024 map. We've seen these all over Long Island. But in 2024 it was largely on the South Fork, associated with septic systems and also legacy fertilizer and agriculture. But again, emphasizing the nitrogen front, if I was giving this presentation maybe 2 decades ago, I would say, these are being driven by phosphorus. But there's been a real shift in the global science in this, recognizing how important nitrogen is for these events and for their toxicity. I showed you that toxic compound before, microcystin, and what you can see here is this is a very nitrogen-rich compound, 10 nitrogen atoms per molecule. And we can literally, we've published on this, we can turn on and off the synthesis of this toxin with the amount of nitrogen we give this algae. So if we starve it, it actually, at a biochemical level, it turns off the genes from making this toxin because it has to worry about other things. But conversely, if you give it lots, it turns on the genes and makes more of this toxin. And that's for microcystis and microcystin, but we see that in other harmful algae as well. Moving on to the marine systems, probably the harmful algal bloom that we worry about the most is alexandrium because it makes saxitoxin. That's a neurotoxin, and it can get into shellfish and lead to the human health syndrome paralytic shellfish poisoning. And in some cases, for example in Alaska, that can be a lethal outcome. We've seen alexandrium widespread all across Long Island, and I don't know how appropriate it is, but for this particular map, in addition to showing the density of cells we're seeing, I'm showing the locations that have had shellfish closures due to alexandrium during the past 15 years. And you can see they're widespread, again, a lot on the east end, but there's actually 10 different ecosystems that have experienced these, and actually 36 individual closures since 2006. Now, in happy news, we didn't have a shellfish bed closure due to alexandrium, or actually any of the halves in 2024, but we did see high levels in several systems. And historically, we've seen that nitrogen can really trigger these harmful algal blooms. So in this particular figure, what I'm showing you is an experiment. I may show more of these later, but what you can see in the experiment is essentially capturing this bloom water and incubating it either with nitrogen or phosphorus or both compounds. And in just 48 hours, we can elicit a 2.5-fold increase in the densities of these algae just by the presence of excessive nitrogen. And that's not a surprise. We've published on this in the past, a couple of papers over the years, making links not just to nitrogen, but specifically wastewater-derived nitrogen. When we've been able to track the nitrogen that we find in the cells, in the alexandrium cells using isotopic signatures, it's a very clear wastewater signal, not from fertilizer. Another marine harmful algal bloom we think a lot about is Dinophysis acuminata. This is one that makes something known as oxalic acid and some related toxins that can lead to diuretic shellfish poisoning. And this is actually even more widespread in alexandrium. We've actually yet to find a location without it. So it's always somewhere in the background there. So that leads to the axiom in microbial ecology: everything is everywhere and the environment selects. This organism is everywhere, and so the question is, when will it grow dense enough? And the answer is when the environmental conditions are right. We had a surprise event last year in a place called Little Seatuck Cove. I feel like sometimes I think I've monitored all the water bodies in Long Island, but this is one I've never seen before. We had sampled Seatuck Cove, but not Little Seatuck Cove. But I had a former student contact me by cell phone and send me this picture and said, I think you should take a look at this creek. Something looks off. And in fact, it was off. It had dinophysis, but at this enormously high density of 100 million cells per liter. And for perspective, before that, the highest we'd ever seen in New York was 2 million cells per liter. And I would say that, that's my personal observations, but I do work with global colleagues. And so in being in contact with really the global expert, Beatriz Rivera from Spain, she's convinced this is the highest level she had ever seen. Last year I published a paper with colleagues from the West Coast, from the Gulf Coast, from the East Coast, and individuals from NOAA, and all those locations. We compared dinophysis blooms across the country. And this again, the 2 million was higher than anyone had seen anywhere. That paper was published in 2023. But here we're now 50-fold higher than that. The bloom stayed above 1 million cells per liter for almost a month. DEC closed this, precautionarily closed this area to shellfishing, and we had collected clams and sent them to the Virginia Institute of Marine Sciences, and indeed the clams that were collected there were well above the federal limit. Some of these graphs came out a little bit different than I would have thought, but an interesting thing is we went back to Little Seatuck Cove this year. We figured, okay, this was an enormous bloom. We'd never sampled this before. Let's be ready this year, and let's not be surprised, and let's really try to understand what's leading to this bloom being so intense. So we got out there nice and early in the year. We started sampling, and instead of getting 100 million cells per liter, it got to 2,000, and that was it. So these blooms seem to move around. However, we do monitor a lot of places across Long Island. To our surprise, the bloom wasn't in Little Seatuck Creek, but instead just 8 miles to the northeast in a place called Reeves Bay. In the Peconic Estuary, we saw very similar sort of bloom with regard to intensity at 60 million cells per liter. And I'm not sure how many of you can see the image there, but that's a microscopic image. And this is, it's not monoclonal because we know there's lots of little things in there, but with regards to the big phytoplankton, this was the only thing in the water, which was really surprising. And again, just yesterday I met with colleagues, again, from the East Coast, the Gulf Coast, the West Coast. We're all focused on Dinophysis. And this was something that took a lot of everybody on that Zoom by surprise. And this is the progression of that bloom getting to that 60 million cells per liter. And so it comes in as the second most abundant. But again, if I was giving this talk 2 years ago, it would have been, we would have said 2 million is as high as it gets. This was above 2 million for almost a month. And I'll just point you towards these 3 different papers we've published over the years that have made very clear links between high levels of nitrogen and the occurrence of these blooms. We've shown it in an ecosystem setting, we've shown it in the laboratory, the organism, and this was, you'd think it would be true for all phytoplankton. For this one in particular, I don't have time to explain why. People originally thought that, maybe it doesn't require nitrogen, but actually it does, and we've tested multiple strains isolated from locations in the East Coast from as far north as Maine to as far south as Virginia. All the northeast isolates, more nitrogen makes them grow a lot faster. Okay, moving away from the harmful algal blooms, I want to talk about oxygen. I talked about dead zones earlier. And the overall paradigm here as to how we get low oxygen does link back to nitrogen. That the nitrogen stimulates harmful or algal blooms in general. That creates a lot of carbon in the ecosystem or organic matter, and when that's broken down, that pulls the oxygen out of the water. And through the Clean Water Act, it's one of the few water quality parameters that have standards that are monitored and regulated by DEC. And so specifically, the standard would be that we want about 4.8 milligrams per liter of oxygen in the water, but we never want it to go below 3 milligrams per liter. And that's to support fisheries. And so we monitor a lot of places across Long Island, and we've been doing that for a long time. And so this past summer, if we look at the oxygen levels, you can see this map from behind, anything that's red is less than 3 milligrams per liter. So when it gets to summertime, a lot of these places, or most of these places, are not reaching that standard. And almost none are making the highest standard. And then the question is, what are the implications, right? These standards were meant to protect fisheries. If you read the language, and I feel like I have it in here somewhere, or maybe I put that somewhere else, but the language is about supporting fish, shellfish, and wildlife. So does the oxygen matter? And we specifically did some experiments to test this recently. One of my graduate students did, where we took water that we knew would be lower in oxygen just at night. So not really, really bad, but having nightly excursions of low oxygen. And we took that water in, we vigorously bubbled it, and then delivered it into large vessels where we can continuously monitor the oxygen and look at the growth of shellfish. And so here you're looking at the oxygen levels that come into this place, Old Fort Pond, where the Marine Lab is in Southampton. And you can see a day-night cycle, and the levels are good by day, and they're low at night. And this has actually created this paradigm whereby, for decades, the mantra was, the oxygen, there's no oxygen problem. We go out every day, and we measure it, and every day the oxygen is high. And that was correct. But the problem is that as soon as the sun goes down, the oxygen levels start declining, and it can happen quite rapidly, particularly in summer. But again, this is not by any means one of the worst sites in Long Island. In fact, it's just moderate. You can see sometimes at night it goes below that standard. But by bubbling, we could bring that oxygen level way above that standard. So the question is now, let's just grow up the shellfish and see if they care. Does it matter? It turns out it does. So now, all the shellfish we use, again, I apologize, some of these graphs moved as it went from a PC to a Mac. But this is essentially showing the growth rate of the oysters. And so growth rates of the oysters in this particular case increased by almost 50%. And we could, I lost the figure there. Things are getting worse with time. The next figure that I would have shown you is that we did about 10 of these experiments. We used clams, we used oysters, we used scallops. And we could create a very tight linear regression between how much their growth rate would improve with oxygen that was related to the amount of time the oxygen was low at night. So all of these species responded as long as the oxygen was low. Because in the beginning, we weren't sure what was happening. We had some experiments where their growth went up, and others where it didn't change at all. But when we looked at what the actual oxygen levels were, it was very clear. The ones where they didn't respond is because it wasn't going below 5. Every time they're having that nocturnal hypoxia, their growth rate slowed. And this does matter. This plot here is showing you the survival of clams based on their size. And so essentially what it's saying is that the smallest clams, 10 millimeters and smaller, Carl and I were just talking about this earlier, just don't survive in an ecosystem setting. They're like chicken McNuggets for crabs. They love them and they eat them up very quickly. And it's the bigger they get, the more likely they are to survive. And that big bar, of course, is for the adult. And so if shellfish are experiencing hypoxia, low oxygen at night, and their growth slows, it's basically going to take them much longer to get to what we call the predation refuge size. And so it's going to translate not just to their slow growth, but ultimately that'll be to their mortality. Okay. And now, just briefly, I want to touch on the idea: is this really a connection between nitrogen loading and algal blooms? Does that kick off algal blooms? Does that lead to low nitrogen, low oxygen? So to answer this, we performed experiments across 25 locations all across Long Island. And we collected water, and we conducted a fairly straightforward experiment, similar to the one I showed you before. We collected the water, we amended it with nitrogen or phosphorus or both compounds, and after 48 hours, we looked to see how does the algal growth change. Does it change at all or not? And then we did a second follow-up experiment, same vessels, put them in the dark, and said, did the oxygen consumption rate change or not? Pretty straightforward. Now, I'm not going to go through 25 different experiments, but I'm going to give you the upshot. And again, this is another figure that somehow got cut off a bit, but the first bar I'm showing you here is what percentage of the experiments did nitrogen significantly increase the growth of the phytoplankton? The answer is 92% of the 25 locations. And what percentage of experiments did adding nitrogen decrease the amount of dissolved oxygen? And the answer is every single experiment. And then how much did the nitrogen change algal biomass? Increased it by 260% on average. How much did it change or decrease the oxygen levels? By about 110% on average. So, I don't see how these experiments were all done in the summer. That's one caveat I'd say, but I can't see the experiments being any more cut and dry and clear than this. Okay. I want to pivot now just briefly and talk a little bit about climate change. Of course, many scientists, or some scientists argue we're in the Anthropocene, the era where the activities of man are overwhelming the natural forces of the planet. And as we look at what's happening locally, we really do need to consider what's happening globally. And the signs of climate change are all around us. 2023 was the warmest year on record, and this was like by a long shot. We'll see where 2024 lands, but it's going to be close. If you didn't know, we had back-to-back days this summer that were the warmest days ever on planet Earth. So this is one of those 2 days, July 21st, 2024. There's a great site called Climate Reanalyzer run by the University of Maine, and there's probably a few other people besides me who are like this, but I check this site a lot, many times a month, because it gives you, by the day, what's the temperature of the global ocean, and you can actually break it out by location. So here I'm showing you the temperature of the North Atlantic, and all the little gray lines are years in the past, and in the yellow and the red are 2023 and 2024. Remember, 2023 is the warmest year ever on planet Earth, and this is our oceans, and you can see 24 and 23 are racing back and forth. We're not sure who's going to win. But this is just for the North Atlantic. But like if you look at our region of the North Atlantic, it's true to say that today, on this day, what is today? October 18th. This is the warmest October 18th the North Atlantic has ever seen since we began monitoring. Even warmer than it was in 2023. Beyond that, we know locally our summer water temperatures are increasing at a rate that's more than 3 times the global average just since the turn of the century. We've been using new techniques that can take advantage of satellites. With satellites, you can measure surface temperatures. And so with satellite surface temperatures, you can get a reading every single day per square kilometer. That gives you a lot of good temporal and spatial coverage. And so when we look at different seasons, we see summer really standing out as the time of year that it's getting much warmer here. And as we probably know, when it gets warmer, that can lead to more humidity in the air and more rainfall. So I just want to talk about that momentarily. You may be aware that the trend with regards to heavy rainfall events in the Northeast US is a 70% increase since the middle of the 20th century. This is like those really big torrential downpours that we experience. And you may know where I'm going with this. The event on August 19th that happened here on Long Island, where some areas of Long Island got more than 10 inches of rain, so much so that it blew out the Stony Brook Mill Pond and Blydenburg Pond. They just don't exist anymore. And so this was called the 1 in 1,000 year rainfall event. You can see the headline, and then below that I have a press release out of the governor's office. And again, using that term, the 1 in 1,000 event that exceeded rainfall events of Hurricane Ida. So, and these rainfall events are now happening more frequently. And so just to review then, if it's 1 in 1,000 years, let's go back. Well, there's Hurricane Ida, indeed also dumping close to 10 inches of rain. So very, very close between the two. And then here in the South Shore, I have to imagine there are people in the room who may remember the 2014 event. Now that was the record event. And I don't know how people forget this, but this was the all-time record for rainfall in New York State. 14 inches fell all at once. And here's some data on exactly where that happened. Didn't come up with a name, although the Wall Street Journal called it the super soaker. But 14 inches of rain in Iceland, just in a 24-hour period. And this paper was just published in the last year showing how frequently will we get the 1 in 1,000 year event. And you can see the bullseye where we are. And that's on the left side is showing you the 1 in 1,000 1-hour rainfall event, and on the right side is the 24 event. But either way, we're in the bullseye right here for high rain events. And so, was this year really 1 in 1,000 years? Well, if we had one in 2024, and 2021, and 2014, this is not the 1 in 1,000 year event. This is the every few year event, and that's what this study actually shows. You can see the darkest areas are less than 5 years. That's how frequently we're now getting the 1 in 1,000 year event. And this is important because, as you know, that rainfall is not coming in fully clean and pristine. You saw my very first slide, I talked about the idea of the watershed. One, we know that this rainfall delivers a lot of pathogens to surface waters. I know that shellfishing was closed in many places for more than a week following that event. But it's also bringing more nitrogen. So I'm going to highlight this paper from a colleague of mine from Stanford University, and specifically calling out the idea that, yes, there's going to be more rainfall in the 21st century. And what she highlighted, she called that, of course, the Northeast, where we are. But the fact of the matter is that this increase in rainfall alone is going to increase nitrogen loading by 1/3. So it's going to make things that much worse just with the increased rainfall. And again, the same paper, she called out where this is going to happen. And we're not solely the bullseye; it's both the Corn Belt and the Northeast. But we're the area that's going to be getting more of these heavy all-at-once rainfall events. Certainly, temperature is another big part of climate change. I've already talked about that, and And this was the warmest day in the world until, this is July 6, 2023. This was the warmest day in the world until July of this year. But one thing about warming oceans is that it moves the organisms in the ocean. And that is to say that as water's warm, organisms will migrate towards the poles to stay in their thermal optimum, right? Every organism has a thermal optimum. They want to be where it's just right for them. As our planet warms, organisms are going to start moving towards the poles. And we've seen this for many marine animals. You know, the Long Island lobster is no longer here, right? Why? It got too warm. It's thriving in Maine. So I want to move now to a bacteria of concern in marine waters called Vibrio vulnificus. And this is the patterns of Vibrio cases in Florida and Texas and associated with temperature. So the temperatures are the line graph, the bars are the cases, and you can see these cases get the peaks of these cases are during the warmest months of the year. So what is Vibrio vulnificus? So it's a bacteria that really likes warm water. It also needs brackish water, so it can't live in fresh water, for example. It can infect wounds. Sometimes it can accumulate in shellfish. People who are immunocompromised are the most vulnerable, and it can lead to, essentially, it starts eating up the tissue and human tissue, and it can be very serious. I mean, this is CDC information right off the CDC webpage, so I'm not trying to, exaggerate things, but you can see the outcome. 20% of people that are infected can die in 24 to 48 hours. And there's a climate change link. So this is a paper that came out just last year, and what it essentially was showing, if you look at the lines that are different colors, it shows the migration of this organism up the East Coast from the 1980s till today. And so what it's showing is that it used to be only in the Gulf. Right? And since then, particularly during this century, it's been creeping up the East Coast. So these scientists ran a model to say, well, when will it now become an issue in the Northeast? And so this is a 2x2 map. It's showing you 2 time frames. You can see that. And it's showing you what happens with low carbon emissions and high carbon emissions linked to temperature changes. And so what the scientists concluded is that if we don't curb emissions, by the end of the century, we'll have Vibrio vulnificus on Long Island and New York. And this was published in April 2023, and that was a great paper until 5 months later, Vibrio vulnificus was in New York. So it didn't take that long, it happened already. You can see the headlines here from the New York Times, and I'm going to make an amendment because I don't think this is an oyster issue in New York, and I'll talk about why momentarily. But not only was it the New York Times, but the CDC actually came out with what you can see here, one of their health alert network responses. They put these out as emergencies, and they did put one out last September to look for severe Vibrio infections associated with warming coastal waters. We did a survey last year looking for Vibrio, and in fact found Vibrio vulnificus. So I should emphasize this is a quantitative and digital PCR method we're using. We're using DNA fingerprinting of the organism with positive controls, negative controls. So there's no doubt that based on the genetic sequence, we did find it in different locations across Long Island. The happy news for those of us in the room, just one relatively mild hit in Great South Bay, but some other hits across the South Shore. And 2 spots that really stuck out were some areas in the North Shore, western part of Long Island Sound, and also some areas in East Hampton. Densities were inversely correlated with salinity, so that is, you get those pulses of fresh water, and you have areas of lower salinity. Those are the areas that were most likely to have this organism. It looks like this one also moved a little, but I'll just talk you through it. So this year we've been monitoring as well. I don't have all that data together, but we also did an experiment. We're doing a series of experiments, trying to understand what's driving this and what can make it better and worse. And so this particular experiment, it's one of dozens we've done this summer, but it's consistent with the other experiments. We collect the water, and just like we've done with nutrients, now we're amending it. Instead of with nutrients, we've tried different nutrients and different sources. This one in particular, we actually used on-site wastewater. And it would take me a long time to explain why. But we use on-site wastewater that came right out of a septic tank, that went through a sand filter, and then with both cases, we also filtered that water afterwards. That's why there's 4 treatments. So it's, again, a little shift in the figure, so I'll read it out, I'll say it out loud. It was raw wastewater, filtered raw wastewater, sand filtered wastewater, and then filtered sand filtered wastewater. And so here's the outcome. And frankly, I've never seen a microbe respond like this to anything. A 2 order of magnitude increase in the densities in 24 hours upon exposure to wastewater. And we've tried other things with it. We changed the salinity, organism didn't care. Changed the temperature, organism didn't care. At least in these 24-hour incubations. We give it organic matter associated with phytoplankton, or in this case just organic matter associated with wastewater, and it's an astonishing increase in densities. From a few thousand to millions per 100 milliliters. Now, a few things I just want to say, just because if you take all this in, it could be a little alarming. First thing I'm going to say, we do not have an epidemic of Vibrio vulnificus on Long Island. If we did, we would know, because there'd be a lot of people, frankly, in the hospital and dying. So this is a very, very rare event. And even in Florida and Texas, they're having hundreds of cases per year, not thousands. And we would know if we had hundreds. So I will say, though, I do know people who have had skin infections here. And again, I want to emphasize, and I'll explain why by the end, but I think skin infections is the real path here. This is a natural bacteria, and so in Florida, where they have this a lot, they recommend that don't go in the water if you have a fresh and open cut, particularly in the summer. Makes sense. If you're immunocompromised, make sure you have good footwear. Right? If you're walking on the beach, you wouldn't want to cut your foot and then have it be infected. And then if you do get a cut, make sure it gets cleaned, and if it looks like there's something wrong with it, go see your doctor. And recreational shellfishers mind the rules. I want to say that the DEC has dealt with Vibrio for a long time, so they know all about it, and they've put in great restrictions for another species of Vibrio, Vibrio parahemolyticus, whereby in summer when the shellfish are harvested, they're put on ice immediately. And by doing so, that prevents the overgrowth of Vibrio. So I don't think there's a Vibrio problem with shellfish, but I do think there could be Vibrio issues with regards to skin, and again, I know people who have had these infections. Okay, and again, just summing this up, it's a combination of warming temperatures, more and more rainfall, and the septic systems are clearly not helping. Okay, so now shifting to things that do help and mitigating nitrogen, I want to highlight Long Island Sound. For those of you who don't know, the Long Island Sound study put together a nitrogen reduction plan that went into effect in the year 2000, whereby they reduced nitrogen loading by 60%. That's a huge number. Now that was I won't say easy to do, that was very expensive to do, but because most of the nitrogen loading going to Long Island Sound were from sewage treatment plants in and around New York City, it was just a matter of upgrading dozens of sewage treatment plants. It cost billions and billions of dollars, but they got it done, and they reduced the nitrogen loading by 60%. And what you're looking at here is really before and after, on the left and the right. And that's the extent of hypoxia, or number of years that there was hypoxia before implementing the plan and after. And so what you can see is there's a wide swath of Long Island Sound that used to be hypoxic every summer and is now never hypoxic. In the bad years, the dead zone, this low oxygen zone in Long Island, extended 2/3 of the way across Long Island Sound. And now it's really isolated to the far west. And you can see there's the zone that's cleared up nicely. This is the relationship between nitrogen loading and the size of the dead zone in Long Island Sound. And it's not the perfect correlation, but it's pretty good. And this is from the Long Island Sound study, and I have an arrow to the size of the dead zone in 2024, one of the smallest. Right? And there's a lot of headwinds working against this being successful. Warmer water holds less oxygen. So as our water is warm, if nothing else was done, the oxygen would get worse. But in fact, it's gone in reverse. By reducing the nitrogen load, this dead zone is shrunk to being smaller and smaller. And there was a point in time I thought for this summer that we might not even have a dead zone in Long Island Sound, because it really only happened like during a single cruise. Okay, so beyond that, when we're here in Suffolk County on Long Island, we've got not just a few sewage treatment plants to upgrade, but we've got 400,000 septic systems to work on. And the Suffolk County Sub Watersheds Plan has come up with a plan on how to deal with these and how to prioritize these. And this is a map from that plan. And you look very carefully at this map, but I want you, and just focus on the bright pink areas, right? That's what they're calling phase 2 of this plan. And those are the areas they want to upgrade, where they want to upgrade the septic systems. And what I want you to note is that this is very clearly not a willy-nilly plan. Somebody didn't just say, okay, we'll draw a straight line in every home that's within 5 miles of the shore. Well, no. This was a very deep dive into the science. And they essentially first said, okay, 1, let's protect public water supplies. Let's make sure we're getting nitrogen out of public water. 2, let's look at the water bodies that are most vulnerable to high nitrogen load. And let's target those regions. So you'll note, like on the very north shore of Long Island Sound, not a priority, at least for this phase 2. You'll also note on the South Shore we are here, huge priority, right? Most of the watershed in the South Shore. Now, why would that be? The South Shore is so shallow, and the tidal flushing is so low, it leaves it very vulnerable to excessive nitrogen. And so that's why that's the area that's being targeted for this. And I just want to point out, I just came across this in the last week, but I think it's relevant. If you don't know, New York State is shrinking in population. People are leaving, they're moving south. You can all describe to yourselves why that may be. However, Suffolk County is bucking the trend. So we actually will have more people, we have now more people than we did just a few years ago, because we're one of the few counties that are growing. Nassau County shrinking, the city shrinking, Suffolk County growing. So more nitrogen load than we thought originally. Okay, but Suffolk County does have a plan for upgrading these systems. You can see these are some of the approved systems. That's a really interesting change in the slide. At Stony Brook, I direct the New York State Center for Clean Water Technology, where we're harnessing science to engineer clean water for the protection of public health and the environment in New York and beyond. And we've come up with an alternative septic system that has provisional approval in Suffolk County. It's called the nitrogen removing biofilter. It's made with just sand and sand with wood chips. And exciting news, this is a non-proprietary system. Anybody can install it, any installer can do it, and there's no patents. Anybody can do it, there's no money to be made by anybody except for the installer and the people who sell the parts. So it's a drain system above sand, and then the sand goes into a layer of sand mixed with wood chips. And that leads to nitrification and denitrification of the wastewater. Another slide where things really got muddled there on that y-axis. These bars show the performance of different alternative septic systems in Suffolk County. You just have to look at the colors because of the muddledness of that x-axis. But the ones in red are the 2 nitrogen removing biofilters. So I'm excited to say these are the best performing systems in Suffolk County, and this is Suffolk County data, right? So you can go in and make the same plot yourself. The very far right is an existing system. So you can see about 100 milligrams per liter of nitrogen, we're able to get it down below 10. So we're excited about that. But we also have to think about more than just nitrogen wastewater, things like emerging contaminants. And the systems that we've made actually remove more than 25 different pharmaceuticals, drugs, personal care products, and solvents. And the removal percentages are as high as or higher than sewage treatment plants. And this is actually because of that sand layer. We've actually done the deep dive. We've identified the microbes that are doing this. The microbes are breaking it down, and they're the microbes that attach to sand. So by having that sand layer with the oxygen in it, that's where the magic for this is happening. And again, these have been improved and can be installed. And in some, you can get grants for up to $40,000 on the East End, $20,000 for the rest of Suffolk County for installing these. So beyond addressing the septic issue, we know that it's going to take time to upgrade those 400,000 homes. We also know that it takes time for groundwater to flush out. And so we need what we call in-the-water solutions in the meanwhile. And so there are some exciting things to talk about there. And one has to do with filter feeding bivalves. And this is something I know Save Great South Bay is doing a lot of work on. And I think there may be some signs for Great South Bay right now that this is in action. But the whole idea that by having more bivalves that are filter feeders, they can filter out the algae, produce oxygen, promote the growth of seagrass, and lead to a positive feedback, and keep some of these things like harmful algal blooms under control. And in the interest of time, I'm not going to go through this paper, but if you're interested in this paper, you can get this online for free. And it's the story of the Shinnecock Bay Restoration Program, where we planted 5 million adult clams. Those adult clams were not the filter feeders, but rather those 5 million adult clams, through the awesome power of clam biology, had about 50 million offspring. And over a decade, those 50 million grew up, and then they became the ones that were filtering the estuary. So it's an exciting story of increasing the population, increasing the harvest. We estimate that the increased harvest in Shinnecock Bay, because baymen are suddenly making a living there, is responsible for tens of millions of dollars in the economy. So it's a happy story, and you can invite you to read that paper. And then the last thing I'll mention are seaweeds, and specifically something known as kelp, that you may be familiar with. Kelp is a photosynthetic organism, takes in CO2, takes in nitrogen, and produces oxygen, may even produce things to work against harmful algal blooms. So over the past 5 years, we've grown kelp in my lab at 16 sites across Long Island, 10 of these sites being oyster farms. And the kelp can remove a lot of nitrogen. The first commercial kelp farm is in Mauritius Bay, Sue Wicks's farm. It's called Violet Cove. And she has a 1 acre plot of kelp there. And we estimate that in a given year, that kelp can remove up to 200 pounds of nitrogen. And that's on par with up to 10, or in the proximity of 10 septic systems. Of course, you have to grow it every year, and it's not saying we do that instead, but I'm saying that it can make a difference. And we've done some work on an adjacent farm called the Great Gun Shellfish Farm, where we've grown kelp, and then we've monitored water quality and also monitored the growth of bivalves. So firstly, showing here, when we grow oysters in and around kelp, they grow faster, significantly faster, and this is a repeatable outcome. We've seen this on the North Shore and on the South Shore. And we think partly because the kelp changes the water quality. So this is a mapping we did on the kelp farm, and what it's showing is this, if you will, tongue of high pH water around the kelp. By taking in the CO2, the kelp is raising the pH of the water and protecting oysters against acidification. And you can see that here also now as a time series. Within the kelp, when we put a sensor out, the pH is consistently higher than the areas around it, and we think that's responsible in part for the faster growth of the oysters. I mentioned Alexandrium before, and what happens when you mix Alexandrium with kelp? This is what happens. That's a healthy Alexandrium cell on the left, and that's an Alexandrium cell exposed to kelp on the right. We call that Alexandrium spilling its guts. The kelp releases some sort of compound that is bioactive against Alexandrium and actually some other harmful algal blooms as well. So it may actually protect the shellfish in that regard also. And now just showing you numerically, when we grow, we put kelp into experimental vessels. This is in case 300-liter vessels. We significantly decrease the amount of Alexandrium in the water. And we're now looking at other seaweeds. Kelp is great, but it's a cold weather seaweed. So we're now looking at some red branching macroalgae that we think will do well during warmer months that can provide the same services as the kelp. And with regards to these seaweeds, I call it the halo effect. We're not going to solve our water quality problems with seaweed. But if we put seaweed on shellfish farms, we do think it can have a positive outcome for those shellfish with regards to harmful algal blooms and also acidification and potentially oxygen. So with that, I'm done. Just to wrap up, excessive nitrogen loading from wastewater is a threat to coastal ecosystems and human health. Climate change is accelerating, and lowered nitrogen is having a compounding effect on our coastal ecosystems and economies. Upgrading septic systems is a primary mitigation strategy for reversing water quality impairment and removing contaminants from things we don't want getting into our drinking water. And seaweeds and shellfish hold a promise to mitigate water quality impairments like acidification and harmful algal blooms. So with that, I am done, and I thank you very much for your attention.
So we'll take some audience questions.
How's that?
Okay. Tracy. Hi. So the filtering systems that you were talking about, is that what people are installing when they're installing advanced?
So the one that we have is just one of several. There's been, I think, 6 other ones that have been approved.
Okay.
Most of them are by a company called Fuji Clean and Hydro Action. But that's an example of what they call an innovative and alternative low nitrogen septic system.
And then another question. When you're doing these experiments in your lab and you're adding nitrogen, are you the 25 sites and the other experiments, are you specifically adding nitrogen with that isotope that is wastewater isotope? Does not really matter.
We don't use the isotopically labeled nitrogen. We do, however, use the form of nitrogen in as ammonium, which is what comes in with septic systems. That's the species of nitrogen that you find. Yes.
I had a question on this. I was reading up recently where apparently sewers.
Use the microphone, sorry, in the back.
So apparently sewers typically leak at a rate of 10 to 25% of effluent. So in these studies that you've done determining human isotopes, have you done anything to determine what that would be coming from a septic or cesspool system or from a leaking sewer pipe?
Yeah, it could be in the one particular study that I'm referencing. It could have been either one because they would have very similar isotopic signatures. Yeah.
Rich.
I don't think I need it. I got a brief. Thank you, Chris. This is like my favorite talk each year. It's really good. And for all your work and with your students. Could you tell us a little bit more about the wood chip sand system? Does it not require any electricity? And how much square footage do you need for a typical 3-bedroom house?
Yeah, as far as size, you do need a pretty big area. I want to say like 40 by 20 feet is what is the leaching field, and it can be bigger for bigger homes. And what was the other question? I'm sorry, the size and the...
Does it require any electricity?
Oh yeah, electricity, that's a great question. As of now, the version that we have runs on a single dosing pump. So it's a Home Depot pump, not very expensive. However, we have just come up with an alternative design that runs on gravity. What we know doesn't work is you can't let the system just be at the whim of the house, because then you flood the system and you don't get the removal. But we've actually created something, it's not far off of a bucket, but the bucket fills, and when it's filled, it delivers a dose, and then it comes back. It's not really a bucket, but anyway, we're testing that, and we're hopeful that that's going to be successful. And in that case, it would be run on gravity and no electricity at all. Yeah.
Thank you.
Follow-up question on that. Follow-up question on your filter system. With regards to the wood chips, how often does that have to be regenerated, refreshed?
Yeah. So the way we designed this, there's 2 different systems. One in which the wood chips are physically in a layer just below the sand. There was a second design, I didn't show it, but it's similar. It has the sand layer, but then it goes to actually a box with the wood chips. In both cases, the wood chips are sitting in the wastewater. And when that happens, they're anoxic. And under low oxygen, wood degrades very, very slowly. So, and as an example I often give, and I apologize if people heard this story before, but I once was in Copenhagen, and I visited an old castle, and it was on a fjord, and there was a museum there. And in the museum, they had a Viking ship they had just pulled up from the bottom of the fjord. And the Viking ship was 1,000 years old, and it still looked like a Viking ship. And that's because when wood is in water and anoxic conditions, it degrades very, very slowly. So we know that there are systems that have been functioning for 30 years. Some calculations suggest that wood chips would go even longer than that. The system we have designed with the wood chips in the box are specifically designed as such that if at any point in time you thought that there was a problem with the wood chips, you could access that box and then essentially vacuum them out and put in your septic.
Hey,
Thank you. In November, we're going to be voting on a proposition here in Suffolk County related to the aquifer and sewers and things like that. I was wondering if you could comment on how that will affect things if it passes or it doesn't pass.
Yeah, I mean, I won't get into the political details, but I'll just simply say that that effort, that issue, that ballot issue, will just essentially give the funding and the organization to accelerate the upgrade of these septic systems and to upgrade and expand sewage treatment plants. And that will be an enormous benefit to coastal ecosystems.
thanks, Chris. I just want to say on that, November 1st is typically we do speaker series quarterly, but November 1st, we're hosting a special speaker series before the election on understanding the Clean Water Plan and why voting to approve that funding is really important in order to move the needle on the septic systems.
Thank you, great presentation. Does the improved septic system, will it function okay where you have a very high water table?
Well, I mean, I'll answer that in two ways. First, I'll say presently, and yet another symptom of climate change, of course, is sea level rise. And so presently, a lot of septic systems in nearshore areas are probably more commonly in groundwater than we would like. When new systems are installed, they must have at least 3 feet of separation from the water table. So that essentially creates a reset against, like I said, the system was put in the middle of the 20th century. It's had some rise since then. And we have done experiments with seawater inundation of the wood chips and the bacteria there in the event that something like that happened in a coastal system, and they do fine. So,
Good question. Andy, for the people in the back.
Sure. You, at the end of your discussion, you talked about an alternative seaweed, and you were talking branching red algae. What about ulva?
we're working with ulva as well. I mean, the one thing about ulva is that, well, I think ulva also has a lot of potential. If it overgrows, it can cause ecosystem harm, and so that would be the only caveat with ulva, is making sure that you're not growing it in such a way that then leads to, in an ecosystem setting, overwhelming the system. But we do find that ulva we're also working with ulva as well too, because we think that has potential.
Thanks, Andy. Just for one of our word of the weeks was ulva, and ulva is the leafy green. It looks like lettuce. It's also referred to as sea lettuce.
Thank you so much for speaking and doing all this research, especially because we're young and we need people doing this. I just have a question about if this kelp does really well, and if we do have commercial success, do we think that we could have a biofuel industry on Long Island?
That's a great question, and it's actually a key question, and that is what will be done with the seaweeds? Right? Some of them can be used for food, but that's not going to be the primary use. We actually have done a lot of work and research looking at it as a fertilizer, and we can actually get plants to grow with kelp particularly kelp mixed with other seaweeds like ova, we can get it to grow at the same rate as we do with a miracle grow, for example. So we know there's promise there. Not a businessman, so I don't know how competitive it's going to be with commercial fertilizers. That's going to have to be part of it. But yeah, there's a future in biofuels. Even more exciting, some of the other exciting things from it are bioplastics. People make literally, there's a plastic that can be made from seaweeds that hold like, what did I hear about? Holds olive oil. You spread it on your salad dressing, and then you can eat it. Because it's not plastic, it's actually seaweed. And so there's a lot of promise out there. I think but it gets into the field of economics. You know, how do you create an economically viable industry?
All right, thank you for coming again and offering all of your results of your research. And it's very intriguing to hear a lot of the conditions, the nitrogen loading and the hypoxia events around Long Island. I'd like to focus on Great South Bay, our backyard here for a sec. About 6 weeks ago, 2 months ago, there was an article that was covered in the New York Times, and it was also the study of the DEC, the New York State DEC, and USGS. And it highlighted a critical drop in our aquifer levels. And knowing that, you know, this is a lagoon, estuary, which relies both on salt water from the inlets and also the groundwater infiltration, a high amount of that. So that has been diminished. I think in the Melville area, the center of Long Island, the aquifer has dropped over the last many decades about 20 feet, which would, if you consider the volume of that, it would account for about 10 times the volume of Great South Bay. Is not there anymore. And it's a much higher level. Gravity is bringing it down and essentially providing the perfect environment for clams and oysters and so on, and other shellfish. So it's not there anymore. Do you have any comments on how this diminishment of our aquifer has potentially increased our nitrogen problems, hypoxia, et cetera?
I don't have a lot of deep thoughts on that in particular. I mean, the one thing I will say is I do know in the issues in Melville are bad, but in Nassau County they're really bad. So in Nassau County, they're really at a nice edge with regards to water supply. So that is, what's coming in is balanced by almost what's being drawn out. There is excess, there is discharge, but they're very, very close. And of course, you know, Brooklyn and Queens used to rely on their groundwater also, and they had to stop doing that because they overpumped. In Suffolk County, we still have an excess. There's been a great diminishment, but there is still an excess. You know, one result would be the change in salinity. So the salinity historically was probably higher in the bays than it is, lower than it is now. But the salinity, I think, is still adequate for shellfish. But I need to think a little bit more about, and we can talk more about other implications as well.
Hello. It's good to see you again, Dr. Gobler. My question was along similar lines there. You mentioned in your talk how warmer water has less capacity to hold dissolved oxygen there. If we were able to get our groundwater levels back to historical levels, would that not increase dissolved oxygen capacity in the bays and provide for a better environment for shellfish?
It wouldn't change the temperature. Maybe near shore it would, but only outside of the spots where it's directly discharging, it would not change. Just based on the volume of that water relative to the whole volume of the bay, you wouldn't see a change in water temperature. So very, very near shore discharge points, but not beyond that.
Thank you.
Any other questions? Oh, sorry, Kamiko.
Hi, thank you so much for your talk. I have a question. You were talking about the symbiotic relationship of sugar kelp helping oysters. Is it also in the reverse, that the oysters help the sugar kelp grow?
There could be to some extent. As bivalves grow, they will of course create feces and pseudofeces that could, be therefore depositing some of those nutrients to the bottom that could be, or into the water that could benefit the kelp. So yeah, it's definitely would go both ways, but I think the shellfish get the greater benefit.
Any other questions? I had a quick question for Dr. Gobler. We know that like we've seen very clearly on your maps, that it's really along the shore that is mostly influenced by what happens on the mainland. We say at Save the Great South Bay that it's the mainland that's sick and the bay is just a symptom. So can you talk to how the mainland influences the bay on this side, but are the positive influences when you have the flush from the ocean, particularly over by the Sexton Island area where the oyster farms grow?
Yeah, I mean, it's so crystal clear. Like I said, we monitor 50 sites and probably across Long Island, but we monitor the South Shore. Yeah, if you want to find the best water quality on the South Shore, just go to the Fire Island Inlet, the Richards Inlet, Shinnecock Inlet, Jones Inlet, and yeah, the ocean water coming in is cooler, clearer, lower in algae, lower in nutrients, and and I think that, to my knowledge, in Great South Bay, the scallops know it too, because the scallops are starting to grow in the area just outside of the inlet. So.
Yeah, one of the projects that we are working on is the potential reopening of the Gilgo Inlet. Do you think this would have a positive impact on shellfish in that area?
Yeah, on that front, I always say, I would say be careful. When the Sandy opened the new inlet, or old inlet, someone asked me what's going to happen. I said, oh, that's going to clean out Great South Bay, and everything's going to be great. And what we saw is that it cleaned out Bellport Bay, and that there was great tidal flushing to the east, to Narrow Bay, and towards the Mauritius Inlet. But we actually saw the middle of Great South Bay got a little bit more stagnant than it had been before. And so, there's only, when you open a new inlet, it's going to have to share the wealth, so to speak. So you may end up having less flushing in other areas. And so, yes, the area, if you created a new inlet, the area right by that inlet will suddenly have much better water quality. However, it's not clear what the trickle-down would be, and you mentioned the areas to the far north being the most vulnerable, right? You may decrease the extent to where these creeks and rivers get flushed tidally, because now you've got, it's all concentrated in that one particular area. So theoretically, it sounds good, and yes, in the direct vicinity, it will be good, but recognize there will be other ecosystem consequences and outcomes, some of which we can't predict. So, yeah, I feel like the next thing I was going to say is I feel like I'm in favor of letting Mother Nature decide where it wants to put in an inlet.
Right.
Yeah.
Almost every year, Mother Nature tries to put it in.
Yeah.
Are there any other questions?
With respect to your kelp project, what is the water depth, the maximum water depth that you look to for putting in, say, for lack of a better term, seed kelp? Or...
Yeah. That's a great question. When we started, we were told that it would only grow in 20 feet of water or more. And my colleague, and I should mention Mike Dole, who works in my lab, he's the associate director for restoration of shellfish and seaweeds, and he gets all the credit for kelp. If it weren't for Mike, I wouldn't be talking about kelp today, because he brought it to my lab and said it's something I think we should try, and I said okay, and it's been a great success. He created a new way of growing kelp. Instead of using deep water with buoys and deep mushroom anchors, he was an oyster farmer himself for 10 years before he came to my lab, and he knew about growing things in nearshore waters. He created these stake blinds. I forget what he calls it, 2 toothpicks at a line or something like that. He can grow it in 1 foot of water, and he can get a 12-foot kelp blade out of that. And that's what we do in Mauritius Bay. That's 1 foot at mean low water, but still, it works well or fine in shallow water.
My name is Mark Nuccio from Long Island Boating World. Over the years, I've written many articles about these inlets. Now, that would not be a new inlet. There was an inlet there. There was an inlet at Muncie Island Inlet. And Zacks Bay was originally the High Hill Inlet. And all those inlets fed that area of the Great South Bay. Robert Moses closed them off, and then to the north in Amityville, Cote, once that happened, over the years, the oxygen just depleted and died. So I don't know if, I've been a proponent of opening all those inlets with tubes, etc. underneath. I see that you're a proponent of it too, but I just wanted to state what I have found. Those are just old, they were inlets.
Yeah, in fact, I think the number is over the last 300 years, there's been more than 24 inlets across the South Shore bays that have opened and closed.
Kathy?
Thanks. I'm really intrigued by the design of your filtration system for the new septic system. It seems surprisingly simple. And I'm just wondering if either you could speak a little bit more to how it actually works, or is there a paper I can go to? Is there something on your website that explains? I mean, it's sand and wood chips. That's it.
Yeah, there is a paper we published on it, both in the peer-reviewed literature and otherwise. There's sort of how-to manuals now. I've got what we call a guidance document for installers if they want to install one of these. But yeah, it's a drain field, which is the most common way on-site wastewater is distributed across the U.S., but not in Suffolk County. And then gets a dose of wastewater every hour, depending on what's in the septic tank, how much, that dictates how much. And it trickles through the sand, which permits the nitrification and, again, the degradation of some of those organic contaminants. And then when it gets to the wood chip layer, because it's enclosed, there's zero oxygen, and that leads to what's called denitrification, and converts the nitrate into nitrogen gas. But yeah, I can point you towards the papers. I'm the lead author. There's an internal called Ecological Engineering in 2022, I think. It's free online.
Great. Well, thank you very much, Dr. Gobler. Before we wrap up today, I'd like to just remind you of our speaker series on November 1st. You can register for that on our website or on our social media. It's through Eventbrite. And also would like to give a really great shout out to our sponsors of this speaker series for 2024, Lessings Hospitality for providing this amazing venue and all of our goodies, and the New York League of Conservation Voters who has supported this program for 2024. I'd also like to announce the winner of our raffle. It is Michael Chonch. Is Michael here? Maybe I'm not saying the last name right. Looks like Choncho, 47 West Lane in Bayshore. Oh yeah, okay, all right, great, congratulations. Congratulations. Also, thank you so much for joining us. I would like to ask, before, I'm sure you'd like to ask Dr. Gobler some personal questions. Not personal, but some individual questions, but I would like to get our photo shot done. So I'd like to ask any of our partners who are in the audience who'd like to be in the photo for our website to join us up in the front. We'll do that right now. And anything else? No? And thank you very much for joining us today. Thank you.